What's up, everybody? How's it going? I know you all are thinking, whoa, you got a haircut. I know. My hair was just getting way too long. Had to go. It was getting too hot. Anyway, hope everybody's doing well. I'm Josiah Zayner, if you didn't know. And today we are going to talk about DNA and RNA and the central dogma of molecular biology and stuffs like that. So since this is our third week, if you are following along with the molecular biology of the cell, now is the time to go to the next chapter. Yay. So the first 2 weeks we spent on cell biology, and this week, the next 2 weeks, we're going to spend on kind of like the central dogma of molecular biology, which is DNA and RNA and proteins. This should be in Molecular Biology of the Cell, Chapter 3. So check that out. Molecular Biology of the Cell, Chapter 3. And, read as much as you can. If you don't have time, I totally get it. But try to read as much of that as you can. You'll learn a lot. DNA. What is DNA? So we've talked a bit about DNA and a little bit about RNA. And we know DNA is the building blocks of life, right? So DNA is composed of 4 bases, A, T, C, and G. And those bases, when strung together in sequences, can make proteins. And proteins are what make everything living. Today we're going to focus mostly on DNA and RNA. Now, there's like 4 major macromolecules that we talk about in biology, which are DNA, RNA, proteins, and lipids. Now, what I mean by macromolecules is, there's a lot of small molecules around doing stuff. But in terms of, bigger molecules, usually we focus on those 4. Now, RNA and proteins are the only ones that can technically have separate functions. So, not only can RNA be a carrier of information, right? But it can also function enzymatically. There are such things as RNA enzymes. This is because RNA's its chemical nature, its single-strandedness allows it to bend and move in ways that DNA does not. So DNA is primarily in the cell a way to store information. Obviously, information that's passed on generationally, but also things like VDJ recombination, what they do, right? That's when your body recognizes a foreign invader particle antigen, and then it stores that information in your cells, like a vaccine. When you get a vaccine, your body stores that information in your DNA, and it has a sort of memory. So a DNA is a way to carry and store information. DNA by itself, I would say DNA and RNA are probably the most disposable. I don't know if you could get by with just DNA and RNA as an organism. You, I don't think you could get by without proteins. Proteins are pretty complex and they do a variety of tasks and can form a variety of structures that are really complex. Um, yeah, so the central dogma of molecular biology. So I made a little video. Let's see if it works. Um, woo! I didn't actually make this video. I just added Wow, that was fast. So we got the central dogma of molecular biology, right? So what we have is DNA turns into RNA, turns into proteins. Now, we have a sequence of DNA. Why is there a U there? Gosh darn it. Can't edit this. Let me change this real quick because I want to. Text box right here. T. Bold. Arial. That looks like Arial, doesn't it? Isn't that what font it looks like? Let's give it a background. Highlight. Oh, don't let me choose white. Anyway, this U should not be a U. There's no U's in DNA, right? That's RNA. That's fucking bullshit, man. It's bullshit. How do I edit the background on this? Format, shape. Fill, solid fill, white. There we go. We don't need a bold, right? Let's make it like 20. There you go. No more U. All right. Figured that out. Sorry about that. So, DNA is a storer of information. Now, DNA is double-stranded, but what happens is that, as we saw in the video, DNA is split apart and converted into RNA. This is called transcription. RNA, which is mRNA in this case, messenger RNA, the messenger RNA is then, through translation, converted into a protein, and proteins are composed of amino acids. Now, the way mRNA is converted into protein is through translation, but it's through this massive riboprotein RNA protein complex called the ribosome. The ribosome is huge. Actually, the nuclear pore complex might be the biggest structure in the cell, but the ribosome's up there. It's one of the biggest structures in the cell. It is, we can check. So the ribosome isn't made of just one protein and one RNA. Ribosome, let's just check the ribosome wiki. I'm sure it has all this information about it. All right. So how many pieces are the ribosome composed of? All right, bacteria is a lot. 31 ribosomal proteins and over 3,000 nucleotides. For the whole thing, it's 52 proteins and about 4,500 nucleotides. For eukaryotic, so like similar enough to human, are Norvegicus, that's rat, I think. We're looking at about 82 proteins, and you're talking about 7,000 bases of RNA, right? So this thing is huge. It's huge, right? I don't think it's big enough to see with the naked eye, but it might be, it might be pretty close. It might be getting up there. So, but the ribosome is what creates proteins. So the first thing the ribosome does is it binds mRNA. Now, mRNA codes for amino acids. Amino acids are what make proteins, right? Now, this is a amino acid codon wheel. I don't like that. Let's see. I like this better. So what we have here is we have the triplet bases that code for each amino acid, right? So in RNA, each 3 bases code for a specific amino acid. Now these are just the 3-hand short for each amino acid: serine, proline, threonine, alanine, valine, isoleucine, leucine, phenylalanine. We'll talk more about the proteins next week, but they code for each amino acid. So it's kind of like a, computer program. It's telling the ribosome what to put in this exact situation. Each of the amino acids have different characteristics, so they give proteins different function, right? So what we have is we have tRNAs. These tRNAs, they match up to these 3-base triplet, which are called codons. And when they do, the ribosome attaches the amino acid that's attached to the tRNA to the growing chain of amino acids. So like in this case, we have amino acid for glutamate, which was brought by the glutamate tRNA, which has CUC on it, which matches on the RNA, the mRNA, GAG. Now if we go back to the code and we look for GAG, we see down here, we got glutamate, right? That's what it codes for. So this process just happens over and over and over again. And what we get at the end is a protein. Yeah, and proteins are one of the main functional things in the cell. They do a lot of the cell work in keeping it alive, right? So we got a gene that turns into mRNA through transcription, and then we have mRNA that turns into protein through translation. Now, remember, DNA is just kind of a message storer, while RNA can also function enzymatically. Ribozymes. So ribozymes are RNA molecules that actually have enzymatic function like proteins. They can do all sorts of crazy stuff. And you can actually engineer ribozymes from scratch. A lot of people have done this. Now, one of the main reasons people think that all biologics started in an RNA world, so like people think that before DNA and before proteins there was just RNA, because RNA can both store information and can also function as an enzyme. But I don't think that's completely true. I'm against the RNA world hypothesis. I think it's a protein first world, but that starts to get complicated. But yeah, because RNA can function enzymatically and store information, a lot of people think, well, I don't know actually how many people think anymore, but people think that means it could have been the first macromolecule. It could have led to the first life. So the first life perhaps was an RNA-only life form. Now, there are viruses that have just RNA. So, possibly makes it so that there's a little bit of evidence that the RNA world is true. But, let's see. But, let's be on Team Protein world. We'll talk about that. Well, maybe we could talk about it now, because why not? So, the whole universe is composed of molecules, and there are a lot of molecules out there, right? But the molecules that we know for sure that exist are amino acids, and proteins are composed of amino acids. So when you just look off into interstellar space, just floating around in space, there's amino acids. When they collect meteorites, there's always amino acids in meteorites, right? So amino acids are a molecule that came around probably shortly after the Big Bang or whatever happened in the world started, right? That's because they're extremely simple molecules. So if you look at amino acid glycine, for instance, let's see if we can pull up glycine. So glycine is basically composed of ammonia and carbon dioxide, right? Those are very common in the universe. Very, very common. Some of the most common molecules in the universe, right? So, glycine, an amino acid, you can imagine that it has been around for forever, right? It is composed of some of the basic building blocks of our universe. Now, if you look at, let's check out nucleotides. Without the phosphate backbone. So let's go nucleoside. All right, so here we go. Here's the structure of DNA and RNA bases, right? Look at that. They're complicated as shit, right? Now you're saying, all right, which one do you think came first, right? Do you think glycine, which is this, it's just a ammonia and carbon dioxide, or do you think these crazy things came? Like, look at that shit. That's some complex chemical reaction to naturally create nucleotides, right? It's pretty complex. So, the other thing is that like amino acids can just also spontaneously form into peptide chains. So here we go. We got amino acids that can form into peptide chains. They can fold into enzymatic structures that can do really complex things. And saying that they can't store information, I think that's just a little naive. There's no reason proteins can't store information, right? So I would say that it's highly more likely that proteins and amino acids are the building blocks of life and not RNA or nucleotides. Right? Anyway, that ends my rant there. Back to DNA and RNA. So, DNA can be, it's the size of our genomes and our DNA are not necessarily correlated with the complexity of the organism, right? But the complexity of the DNA might be technically. Eukaryotes have way more complex DNA. So eukaryotes are like, higher organisms, mammals, animals, plants. Whereas prokaryotes, which are bacteria, have less complex DNA, right? So DNA in eukaryotes has a thing called an intron. Now, what an intron is it is a piece of DNA that does not code for a protein. So, in DNA, there are spaces that don't code for proteins. Now, why would you want this? Because sometimes you're trying to store extra information, right? And this information can be complicated. It can be a number of things. You have to understand that, like, DNA is not just chemical, but it's also spatial, right? So the distance between pieces of DNA is also important, not just the sequence, the modifications on DNA. So, right, DNA can be modified chemically to have something that's called like methylation or other epigenetic modifications. Maybe you've heard of epigenetics. And these things will affect the gene or the expression of this gene indirectly, right? So not everything in DNA is so direct on the gene. Not everything in DNA makes genes, right? So the amount of DNA that actually codes for proteins is, I don't know, let's look it up. Amount of DNA that codes for protein. Codes for glycine. All right, let's see. All I know is it should be a lot smaller than what we imagine, right? God damn it. It should be a small percentage. Where am I? I don't know. How much DNA codes for protein? Let's see. Tell me. Oh man, it's a video? Come on. Just give me a number. What percentage of human DNA is non-coding? Let's check that. Only about 1% of DNA is made up of protein coding genes, right? Now think about that. Let that, so like all the DNA in your cells, the majority of it is not making protein, but it's doing something to regulate the expression of protein, or it's creating RNA or something like that. Now, there are functional RNAs we talked about, and it's not just ribozymes. So there are functional RNAs that do a lot of different things. So if you look up the types of different RNA, there are, what does it say? Oh gosh, some of these things say there's only 3 types of RNA, which is totally not true. All right. So there are messenger RNAs, ribosomal RNAs, signal recognition RNAs, transfer messenger RNAs, small nuclear RNAs, small nucleolar RNAs, mRNA transplicing, small Cajal body-specific RNAs, guide RNAs, ribonuclease P, ribonuclease MRP, etc., etc., etc. There are just a long list of different RNAs that have different functions, right? Now, in most cases, if you're working in genetic engineering and molecular biology, you don't really need to know about all this stuff and about all the different RNAs. Honestly, I have no idea. I'm not keeping up with the number of RNAs and the different RNAs and what they do and all this different stuff, because mostly it's not going to affect you, and mostly these things are more scientific researchy than they will have a direct impact on anything you do. in terms of research or just, life or stuff in general. All these crazy RNAs. You can mainly just focus on the mRNA, maybe the transfer RNA if you want, and if you're doing CRISPR, of course, guide RNA. But otherwise, the whole bunch of different RNA thing. It's good to know that there are different RNAs out there, right? That do a lot of different things. I mean, if the genome is 99% non-protein coding, there are RNAs out there doing stuff also. But try not to get caught up in that. All right. So we talked about DNA and RNA, and let's talk a little bit about epigenetics and epigenetic modifications in DNA. Now, epigenetics was something that was really hot in from about like early 2000s to mid to late 2000s. It was really hot. That's when people first really started putting effort into studying histones and histone methylation and DNA methylation and all these different things, and ChIP-seq and ChIP-chip and all these different techniques, and things got a little crazy. What we kind of came to the conclusion of, though, is that epigenetics is complicated, and it's not as easily understood as some people make it out to be. It's difficult. It has different meanings in different contexts, so you can't always 100% predict the effect of an epigenetic change on a piece of DNA, right? And there are tons of epigenetic modifications, just like everything else, because scientists get obsessive and they like to study the stuff to shit, right? So, epigenetics, the different types of epigenetics. There's direct modification of DNA. Now, usually this is methylation of the DNA, a direct chemical modification of the DNA. And this is known to regulate gene expression, usually contribute to the silencing of gene expression. But yeah, it's not 100%, and it's not always the case. So like I said, epigenetics is more complicated. If you hear people talk about epigenetics and be like, oh, epigenetics is crazy. Like it can do all these things, and I'm gonna modify my epigenetics to like help me live longer or do something, right? Be a little bit skeptical, Be a little bit skeptical, because if there's no evidence that people have shown that epigenetics actually modifies the genes or traits they're interested in, and what they're doing actually modifies these things, be skeptical, right? And then there's histones and chromatin. Now, DNA is not just packed together as like a strand of DNA. DNA has a ton of proteins attached to it. Transcription factors and helicases and polymerases and all these different things. But it also has this protein molecule, called a histone attached to it. Now, histones help DNA pack, and they also kind of circularize the DNA. They take the DNA and they make it so it is more packed, and they also help regulate gene expression. So this is an example of DNA wrapped around some protein histones, right? Now, these protein histones can also be modified on certain amino acids. When they're modified on these amino acids, what it does is it affects gene expression in the cell. It affects gene expression on the genes that it's actually connected to, right? Because you can imagine if you have a histone, right, you need certain molecules like RNA polymerases to come in and interact with the DNA, right? So like if they can't interact with the DNA because there's all these other proteins bound to it, that's going to affect whether that gene is made into RNA and then is made into protein. So histones, as you can imagine, can regulate the expression of DNA. Now, most of the organisms we work with in this course, I think it's bacteria, mostly bacteria. Bacteria have their own form of histone-like proteins, but it's not as complicated and crazy as human and mammalian histones and stuff. Bacteria also don't have introns. Not that it matters much, because when you're doing genetic engineering, generally you do not put introns into the gene that you're trying to put into the organism or into the cell. Sometimes you do, because having an intron helps that gene from not being epigenetically modified. There's something about it. It makes the cell think it's more real. So sometimes people put introns into genes that they put into cells. But in general, you're not putting any introns in. And you're just putting in the gene that you want, and you're having it do the thing that you want, right? So what we could do is we could look at a piece of plasmid DNA. So this is a program called SnapGene. You should download it, SnapGene Viewer. And here we have a gene. Oh wait, you guys can't see it. Let me change this. Here we have GFP, and we have the gene. Now, you have to understand that like genes cannot just be made by themselves. Generally, there's two things every gene needs. One is called the promoter, and that's what tells polymerase to bind there and transcribe the gene. And the other is terminator. That's what tells it to stop where the gene ends, so it doesn't just go and make the whole piece of DNA into an mRNA, right? So you need a place to tell transcription to start and transcription to stop, right? That's called a promoter and terminator. Now, almost all genes are going to have promoters and terminators, and if you're doing genetic modification, the genes that you use should have promoters and terminators. Now, of course, there's lots of caveats to this stuff. There always is. But promoters and terminators is generally just good to use when you're doing genetic engineering. Now, how do you know what promoter and terminator to use? Usually for each organism, there are standard promoters and terminators that people have found to be like, I don't want to say the best, but kind of like the best. So, for instance, this is a yeast plasmid, and it uses a TEF1 promoter and the CYC1 terminator. Now, in humans, what you have is you have what they call the CMV promoter and enhancer, and you have a lot of different terminators, but generally people use something like the bovine growth hormone poly-A signal or polyadenylation signal for the terminator, right? For bacteria, it really depends. If you're using engineered bacteria, if you're trying to do protein expression in BL21 or something like that, you're probably going to use T7 promoter and terminator or something similar like that. It's really easy to find out what these things are. Generally, what you can just go do is you can go find other pieces of DNA or plasmids that have these promoters and terminators in them, and have them, just copy them over to whatever you're trying to create. Now, mRNA also has promoters, right? Generally, in humans, people use the U6 promoter in RNA. And this creates, obviously, RNA so that you can do certain things with RNA. But generally, outside CRISPR, most people aren't using RNA, so I wouldn't worry about RNA too much. But yeah, most of the stuff is pretty standardized. It's not to say it can't be improved on. I'm sure it can be improved on if somebody spends some time and effort actually trying to engineer new and better synthetic promoters for human cell lines. But nobody's done that, so we are stuck with naturally occurring promoters and terminators that work fine for us, right? So yeah. So that is a lot of stuff. Is there any questions about DNA and RNA? Any questions about DNA and RNA? Now, DNA and RNA, mostly what we're just going to use it for is making a gene that we want to have in the engineered organism, right? If you want to make your organism fluoresce, use a fluorescent gene. If you want to make your organism bioluminesce, use a bioluminescent gene or genes. So this week, one of the experiments is to grow up bioluminescent bacteria. These bacteria have been genetically modified to have a piece of DNA that has these genes that makes it bioluminesce. So grow it up, test it out. That's mostly what DNA we're going to use it for. Our 102 class, we go a little bit more in depth in actually modifying DNA. But this class, we're mostly just going to use DNA as a tool. And that's okay also because, it's a good place to start, All right. No questions? No questions at all? All right. If you have any questions, remember, ask them in the Google Classroom. It's good for everybody. Everybody can learn then. Otherwise, I will see you all next week. Thank you for coming to my lecture about DNA and RNA. And have a good one.